Okay, good day. This is Mr. Leventhal again, and I'm going to do a quick summary of the lab that we worked on yesterday. You might recall that we ran out of time, and the end result was I didn't feel like I got to explain with you or talk with you about what we found. So today is going to be, I'm going to try to keep this at a 10-minute discussion about enzymes and just remind you what they are, what they look like, what they do, and then apply that to the lab that we did at the end of the hour. So just a quick reminder to you, when we look at the world of life, life is cellular. And in this case here, we're looking at plant cells. You can tell they're plant cells because of the cell walls. But in addition to having cell walls, you can see these organelles inside. These organelles are chloroplasts. They're membrane-bound structures. And also, you can see that there is a large central vacuole, a water vacuole inside of this cell. And I'll have you think about how that water is maintained in the cell through osmosis and related processes. We looked at this cell. These are allodia. This is what we looked at under the microscope. So if you missed the microscope day, that was one of the cells that we looked at. One of the other cells we looked at were cheek cells. These are human cheek cells. You can tell that these are human cells because they lack a cell wall. And but you can see a nice distinct membrane around them. And we know that they're eukaryotic cells because they have a nice pronounced nucleus in them. And these would be the typical type of cells that you would find in what's called epithelial tissue. These are the tissues that line your mouth, they line your digestive tract, they line your blood vessels. And these are the type of cells that your body uses and kills all the time. Well, why did I bring up cells? Well, of course, we've been learning about eukaryotic cells and organelles in this unit. But really, I brought up these cells because cells have what we call metabolism. And this diagram illustrates cellular metabolism. In fact, you are looking at the complete biochemistry of photosynthesis, which is being represented by this part of the metabolism. And then another process referred to as respiration, which is using oxygen to extract energy. And that's occurring in this part of metabolism. And if I were to name where these things are occurring, well, this is what the mitochondria does. So all of this chemistry is happening in the mitochondria. And all of this chemistry is happening in the chloroplast. Now the rest of the chemistry that you're seeing here, because I don't want to ignore it, is happening in the cytoplasm of cells as your cells are breaking molecules down or making other molecules. And this process of breaking stuff down and making stuff is what metabolism is. And when we look at each of these molecules, if I were to just pick, let's say, this molecule here, and we see that this molecule is being converted or changed into something else, which is being changed into something else, and so on. Each of these changes is being facilitated by an enzyme. So it's enzymes that are helping this chemistry to proceed. And this chemistry is what's occurring in each and every one of our cells. Different cells specialize in different types of chemistry, but this chemistry and the sum of these chemical reactions are the thing that we call metabolism. So metabolism is the sum of the body's chemical reactions and we can divide that up into the reactions that break molecules down. Like breaking food molecules. and the reactions that build molecules up. And this might be like gaining muscle mass, or growing, and so on. Well, it also turns out that when we look at the words catabolism, the breaking reactions, and anabolism, the building reactions, we can also think of them in terms of energy. So when we break molecules down, we are releasing energy. And when we build molecules up, we are storing energy. So this topic of metabolism, or all of the chemical reactions illustrated here, are a mixture of building reactions, in the case of the chloroplast, building sugars, and breaking reactions, and in the case of the mitochondria, breaking those sugars down to create some kind of chemical energy. Well, the thing that's doing all of this biological work are these complex proteins called enzymes. And so this is a protein. Proteins have really large structures composed of many amino acids. And while I'm not 100% sure of what amino acids are where, we could think of this as being where an amino acid is, and this is an amino acid, and this is an amino acid, and so on, all the way along. And so this enzyme that you're looking at might be as many as 400, 500, or even 1,000 amino acids connected together, taking on a shape that does work. And the type of work they do is the breaking of stuff, or the making of stuff. And we can look at this diagram and see that the enzyme is acting on a substrate. And notice that the enzyme matches the shape of the substrate, and that is because enzymes are specific. And that means that one enzyme can catalyze only one reaction. And in the case of this enzyme is catalyzing the catabolic breakdown of the substrate into molecules A and B, and as a result, energy is released. So this might be how you get energy from, let's say, pasta, taking a large complex molecule and breaking it into smaller pieces. In contrast, enzymes can also do building. So here we're going to put two nucleotides together. Nucleotides make up DNA and RNA, and so here our enzyme is polymerase, and polymerase will facilitate the anabolic reaction that stores energy in the new chemical bond. And the enzyme never gets used up. It is recycled and ready to do the chemistry again. Enzymes are expensive for your body to make. Therefore, they want to make them only once or a few of them, and then can reuse those things. Well, let's consider the lab that we did. We did a lab in which we put skim milk into a micro well. We put dairy whole milk into a micro well. We put non-dairy creamer into a micro well. And we put starch into a micro well. And I encourage you to look at the structural formula of the molecules. So dairy products have something called lactose, which is a disaccharide, which means that it is a sugar bonded to a sugar. And here the sugar is glucose and galactose. I don't know what's in the coconut milk, but I know it's not lactose. And then looking at the starch, this is starch, and starch is glucose bonded to glucose, bonded to glucose. And I could keep going, but that sounded pretty boring to me. But it's just a continuous chain of glucose molecules. So we then added to our milk the enzyme lactase, which people who are lactose intolerant can take. And then what we found was that glucose was present after the lactase was added in the two milk products. But we did not find any glucose from the starch or the coconut milk. And the reason for that is the enzyme lactase only works on lactose. The function of lactase is to break this bond right there. And if that bond does not exist, then lactase won't do anything. So lactase works on lactose. We would need a different enzyme to break the starch up. We would need the enzyme amylase, which is in your saliva, and that can break up the starch. So I want to summarize and end this with a few points. And then I'll leave you to study for your exam. So, point 1. Enzymes are catalysts. They speed chemistry. That's point 2. They are proteins. They are coded by your genes. We can always recognize an enzyme because they end in ASE. And then finally, not all environments are perfect for all enzymes. Enzymes have limits. For instance, this graph is showing you how temperature influences enzymes. Low temperatures slow chemistry. High temperatures denature proteins. That means that they break them down. When you have a fever, you feel aches and pains because your proteins are breaking apart or denaturing. When you take an egg and you heat that egg and you go from clear to solid white, this is a denatured protein. The heat breaks the amino acids and the protein no longer functions. Denatured proteins don't work. Final point is just so that you can see that enzymes have optimal pHs in which they function. Some enzymes do great in acids, but some enzymes do not do well in acids, and the same can be said for bases. So in this enzyme here, it's optimized for pH 8. Well, that's it. It's probably 5 minutes longer than I hoped it would be, but hey, at least now we know a little bit about enzymes, and I'll leave you to study and practice. prep for your exam. Cheers. I hope that worked. Oh, what happened to mine?